So we're going to pick up at the end of glycolysis. Now, after glycolysis, your cell is basically going to reach a turning point where it gets to pick one of two pathways. So in one direction, when there's no oxygen in the cell, it's going to go down this route called fermentation. When there is oxygen in a cell, if it's the right kind of cell, if it's a eukaryotic cell, it's going to be able to choose to go into the rest of cellular respiration, beginning with a process called pyruvate oxidation or pyruvate breakdown. Okay, so when we finished glycolysis, this is what we had done. We started with glucose. So that's our carbon-containing molecule, our 6-carbon sugar is glucose. We ended up with 2 molecules of pyruvate. Remember, we basically split glucose in half and converted it to give us some ATP, 2 molecules of ATP on net, and ended up with these 2 3-carbon molecules, 2 pyruvate molecules. The other thing we were doing throughout glycolysis was making ourselves some reduced electron carriers in order to hopefully shuttle them to the end of this process to something called the electron transport chain, where we're going to make a lot of ATP. So in addition to putting glucose into glycolysis, we needed to put in sort of our precursor molecule for this electron carrier. We needed to put in the oxidized form, which is this NAD+. And we're going to make 2 reduced electron carriers, so 2 of this NADH molecule. So we put in 2 of the NAD+. So the glucose and the NAD+ had to go in. The glucose turned into pyruvate. And these important byproducts we got were our 2 electron carriers, NADH, and 2 molecules of ATP for each glucose molecule. So we saw that we actually made 4 molecules of ATP total, but we had to put 2 in to glycolysis started because it's not a spontaneous process. So we had to subtract those from our total yield. Now that's why we only have 2 ATP molecules now. Okay. So an important component of this equation, right, is that we have to put in NAD. And this is fundamental to why and how fermentation happens. So before we get into exactly what fermentation is, and for that matter, what pyruvate oxidation or pyruvate breakdown is, we need to think about what's happening. So for glycolysis, it can happen without oxygen, right? You didn't see any oxygen anywhere in that equation. So glycolysis is happening in the cytosol of the cell, regardless of whether the cell is getting oxygen from the bloodstream, regardless of whether it's a cell even in an oxygen-containing environment. Glycolysis can get the cell energy pretty much no matter what. However, glycolysis is not super efficient. If you recall back on our big diagram of all of cellular respiration, which was a diagram of aerobic cellular respiration, we saw at the end of the process we can make like 30-something ATP. So if the cell has all the components to do aerobic cellular respiration, it wants to do that because that's super efficient. Okay. So after glycolysis, the cell is basically going to check. If it has oxygen, the cell is going to continue with this multi-step process that eventually requires oxygen at the end, and it'll eventually give us a ton of ATP. So the next step of that process is pyruvate oxidation. However, if the cell checks for oxygen and there's no oxygen, the cell is going to have to do something else. Now your cell's going to still need energy, right? Just because there's no oxygen doesn't mean the cell has to stop making proteins or stop moving or stop, replicating. It's still got stuff it needs to do. It still needs energy. And glycolysis gives the cell energy. Not a lot, but 2 ATP is not nothing. So it's going to want to keep doing glycolysis. It's going to want to keep running through that process. However, it's going to come to a stumbling point, which is basically that it's not going to be able to have enough NAD to put into that process. Eventually it's going to run out. Eventually it's going to have converted it all into NADH. So when there's no oxygen, the cell needs a way to make more NAD, to change that NADH back to NAD. And that process is fermentation. So here at the top, we see that equation for glycolysis again. So this isn't an equation for fermentation itself. That's our glycolysis equation. So for fermentation, what we're trying to do basically is coming up with a way that we can turn these NADH molecules back into NAD so we can keep doing glycolysis so that we can keep making ATP. So glycolysis is eventually going to use up all the NAD. If we're doing aerobic respiration, we would run into the same problem, except in our final step, in the electron transport chain, we do convert NADH back to NAD. We do oxidize those electron carriers in order to get the electrons out to power ATP synthesis at the end. So it's not that this isn't something that the cell has to do anyway, but if it's not getting to the electron transport chain, then it needs to do this out in the cytosol. Okay, so formally, fermentation is the breakdown of organic molecules without net oxidation. So what that basically means is that most of the oxidation that happens, so the part where we give away electrons, right, loses electrons, oxidation. In aerobic respiration, so the part with oxygen, that's happening basically at the final step of respiration. So with the electron transport chain, and then a process called oxidative phosphorylation. In fermentation, we don't care about that oxidation. We really just want to keep running glycolysis. So we pretty evenly turn glucose and NAD into pyruvate, ATP, and NADH. And then we just try to turn that NADH back to NAD. So this is kind of a net zero process in terms of oxidation. So this is what it looks like. There are different kinds of fermentation, and that depends on what species you're talking about, rather than like a liver cell versus a brain cell. So we have two types of fermentation. We have lactic acid fermentation, and we have ethanol fermentation or alcoholic fermentation. So an example of lactic acid fermentation is what happens in your muscles. Now, the human body does usually have oxygen running through the bloodstream, so those cells are usually able to do aerobic cellular respiration. But if you're exercising really hard, you use up that oxygen fast. And at a certain point, your cells aren't getting oxygen fast enough to keep doing aerobic cellular respiration. So after glycolysis, when the cell reaches that checkpoint where it checks for oxygen in a muscle cell, at some point, it may reach the decision that, no, there's just not enough oxygen. So it'll switch to making lactic acid in order to keep running glycolysis. So we can see what that process looks like. So glucose keeps getting turned into ATP and pyruvate and some NADH. So those are arrows coming off. One for ATP, one for NADH, and the big arrow for pyruvate. And in order to keep running glycolysis, that NADH has to turn back into NAD. It has to turn back into NAD so that we can feed NAD into glycolysis. And we can't just rip off that hydrogen on its own. We have to kind of run another reaction with it. So what we do is we take our pyruvate molecule that we end up with. We don't need pyruvate anymore. Pyruvate goes into aerobic cellular respiration, but it's not going to produce any more energy for us in fermentation. So we break down that pyruvate, turn it into lactic acid, and that process of basically making lactic acid or lactate helps us make NADH into NAD alongside in the process. So yeast cells, for example, can also do fermentation, but they do it slightly different than a muscle cell. The process is basically the same. They're really just trying to keep doing glycolysis to get those nice tasty 2 ATP molecules. Glycolysis is still producing pyruvate and NADH. We still want more NAD. The only difference here is that instead of turning pyruvate into lactate or lactic acid, they turn pyruvate into ethanol. It's just a different pathway that gets you basically the same thing, except your byproduct here is ethanol, which can be helpful for various culinary uses, right? Like this is how you make beer, right? You put in yeast, you make them do fermentation, and their byproduct of just trying to get normal energy for the yeast cells to work is that they're making ethanol from that pyruvate at the end of glycolysis. Now, going back to our muscle cell. This lactic acid fermentation, or any type of fermentation, doesn't make as much ATP as normal aerobic cellular respiration, doesn't make as much ATP as our final step, oxidative phosphorylation. So if we can go into the rest of our pathway of aerobic cellular respiration, we want to do that. So if oxygen is present, we're going to go into step 2 of cellular respiration. Now this step doesn't itself require oxygen, but it's the point at which we're going into the mitochondria. And the mitochondria, these processes in general, at the end are going to require oxygen. So the cell just kind of knows that that's where it's going. Okay. So glycolysis was happening out in the cytosol. Pyruvate is moving into the mitochondria. Now mitochondria has an outer membrane, has an inner membrane, a space between those two membranes, and then a space inside the inner membrane. Pyruvate is moving all the way through both membranes. So it goes through the outer membrane, goes through the little space between the two membranes, it goes through the inner membrane, and ends up in the mitochondrial matrix. Okay. In the mitochondrial matrix, it gets broken down by an enzyme. Enzyme is called pyruvate dehydrogenase. You can see that on the image. And as it gets broken down, or it's actually being oxidized here, but as it gets broken down, it also ends up pulling off a carbon molecule. So when we pull off a carbon molecule from pyruvate, two things happen, basically. One, that carbon molecule gets turned into CO2. CO2 gets removed. So CO2, that's actually, we're starting to breathe out CO2 that we're producing as we break down pyruvate with pyruvate dehydrogenase. And we're left with a 2-carbon molecule. Basically, we're left with what we call an acetyl group. That acetyl group gets attached to CoA, which is a coenzyme partially made from vitamin A, and it makes a molecule called acetyl-CoA. So we consider that basically one of our products of pyruvate oxidation. Along the way, we're also making one electron carrier, one molecule of NADH. And it's important to note that this is one molecule of NADH for each pyruvate. So in one sort of step of pyruvate oxidation, we get one NADH molecule. But if we were tracking it for one molecule of glucose, we'd multiply by 2. We get 2 NADH for each glucose molecule that we started with. Okay, so here's our equation for pyruvate oxidation. This is showing us the equation for pyruvate oxidation if we started with one glucose molecule. So at the end of glycolysis, we ended up with 2 pyruvate. The 2 pyruvate molecules go into pyruvate oxidation. So we take our 2 pyruvate molecules, we feed in some more NAD. We're going to get acetyl-CoA, which is basically the remaining 2 carbons from each pyruvate molecule. Pyruvate has 3 carbons. We get a CO2 molecule for each pyruvate, so we get 2 of them total. We had 2 pyruvates. We get 2 NADH, so our electron carrier, we get 1 of them for each pyruvate.